So, yes, this is our, oh, it's not wanting to duplicate very well. Let's see. Here, let me just try this again. Now, let's see if this works. There we go. All right, and so lipid biosynthesis is not the exact reversal of beta oxidation. And I was bringing in the notes and set them down someplace along the way. So we've got to then talk about the localization in general. And so where does beta oxidation occur? Since we just covered it. The mitochondrial matrix. Okay? And so it's all about location. And now one thing is that we have compartmentalization that occurs here. And so lipid biosynthesis does not occur in the same location as lipid breakdown, which should make sense, because you wouldn't want to be breaking down and making it at the same time. They're going to be controlled under different circumstances. And so the name of the pathway that breaks down fatty acids, I kind of give it away already, but it's beta oxidation, and that it occurred in the mitochondrial matrix. Where does the fatty acid synthesis occur? In what? I mean, well, what part of the cell does the fatty acid synthesis occur? We're on a very microscopic level here. It's not the mitochondrial matrix. It's in the cytosol. Okay, now we're talking about some of the specializations, like the modifications and elongations and things like that, will occur elsewhere. But for the synthesis of up to 16 carbons, it's going to occur in the cytosol. So this helps with the compartmentalization and the regulation of the biosynthesis of lipids, because we do have endogenous lipid biosynthesis that occurs, even though in Western diet typically we get more than our fair share of fats and the fatty acids from what we eat. Okay, so the very beginning, in the beginning, you have to start with acetyl-CoA. So we're going to see that how we add two carbon units on at a time is with acetyl-CoA. But it's not, if you think back to the breakdown of the even-numbered fatty acids, that was with thiolase. And remember, thiolase can go backwards to where you can put two of them together and make four, the acyl-CoAs. And so we don't utilize thiolase here. We utilize a different enzyme to make malonyl-CoA. Okay, it does require biotin because it's a carboxylation, and it's occurring in the cytosol. One other thing is it requires a different metal than what we've seen normally, and Mn is manganese. And so manganese, by the way, is a heavy metal. It's also neurotoxic, but we do require very, very small amounts of it in a very specific oxidation state. Okay, and then the malonyl-CoA is actually then the starting point for fatty acid synthesis, and 2 carbon units get added on at a time. But this requires bicarbonate and ATP. So what class or classes of enzymes could this be? There's at least a couple of them that we could think of. What's one class of enzymes that this could be? A carboxylase, and that's what this one is. And so if you're going to name this one, what would you call it specifically? And it's acetyl-CoA carboxylase. Okay, pretty simple here. And for those in Organic Chem 2, the chemistry behind this chapter in particular follows really closely with what we're doing in class. With this whole idea of the beta keto groups and beta carboxyl groups. And so for those who aren't in, or it's been a while since you've taken Organic Chem 2, what is so special about the beta, keto, and carboxyl groups?
They can, like if you heat them up, they can cause the carboxyl to leave.
Right, we can do decarboxylations, and they can also activate the carbon that's in between the two, because it becomes more acidic and more reactive than what you'd expect. Okay, and so we're going to see that with the electron pushing in a moment. No, it's just that one, I mean, it's very similar to what magnesium does. It's just the fact that this one is specific for manganese. We're not going to go into like inner versus outer electron interactions and hard metals versus soft metals. Okay. So this is acetyl-CoA carboxylase. In mammals, in us, it's a very large complex, whereas in certain bacteria, the carrier protein may be something that's separate. Okay, so, but for us, there are three functional regions. So we have the carrier protein, which contains the actual biotin group that we've seen before, and it's via a lysine chain, so it's an amide interaction. Then there's a biotin carboxylase region and the trans carboxylase region, and it swings like a little arm. And so it really does, I mean, it specifically tells you exactly what it's going to do. So you can imagine the biotin carrier protein is the one that has the biotin attached to it. That's like the hinge. The one that's the carboxylase is the one that's going to add the carboxyl group to the biotin. The trans carboxylase is going to transfer it to the awaiting acetyl-CoA. So I got this blown up so that way we could see it a little bit better. But once again, we've seen something like this in the reverse with decarboxylations, but now we're building things up instead of tearing them down. So we see the bicarbonate, which, what is bicarbonate used also in our body for? Once again, to review from last semester. It's for pH. We have plenty of bicarbonate. You know, it's from the carbon dioxide. And this is the way that we do our blood pH. So there's plenty of carbon dioxide and bicarbonate around. It does require ATP to activate it. And it gets added directly to the biotin group, which is now going to swing it on over to the transcarboxylase side, where we have acetyl-CoA come in to accept the extra carbon, if you want to think of it, to make the malonyl-CoA. All right, and so that's what the malonyl-CoA looks like. It is a beta-keto acid. Oh my goodness. All right, and so this is all before we can even get started with the actual enzyme that's going to start building up the lipids.
So is that step 0?
Or maybe even step negative. You're gonna find out that it's a lot more difficult to build them up than it is to break them down. I shouldn't say difficult. There's just more steps involved. Okay, so fatty acid synthesis in us occurs in a huge protein. Okay, what enzyme complex synthesizes fatty acids in all known organisms? Does anyone know? What would you call something that's going to make fatty acid? What class or classes of enzymes could that be? There's only two known enzyme names. What family names could it be?
Synthase.
Synthase. And if you're going to call it something, what would you call it?
Fatty acids.
It's fatty acid synthase. It is a repeating 4-step process, but it's not the exact beta oxidation. There is no thiolase. Okay. And this will only make saturated fatty acyl groups, and you should know, like, it can only go for so long. Okay, and then it stops. And they don't actually know by the mechanism with which it stops, like why, like how does this know if I say FAS, that's what I mean, fatty acid synthase. How does it know? We've already taken acetyl-CoA, we've added a carboxyl group to it. Now we're going to have to try to get this huge complex, which I really wish I had my notes so that way I don't miss them out of order. This huge complex that, I mean, it's quite amazing. All right, and so it does require it to be activated. And whenever we say an activated compound, what do we mean when we say activated? Like if something has been activated for, the reaction. What are some of the examples that we've seen? Phosphorylation, acetylation. You can actually have a carboxyl group that could be anything that's going to have a good leaving group, to make it more energetically favorable. Don't worry, we're going to talk about this in greater detail, but this is identical to really the kind of stuff that we've been talking about in organic chemistry last week and continuing on this week, to where this is, I've got a better, more in-depth picture in just a moment. You get the the malonyl-CoA group that's physically attached to a carrier protein or carrier subunit, and it's going to be activated by the decarboxylation. So we see electrons that go and smack into that carbon. Something has to break or leave, and what leaves or breaks is the bond between the two carbons, and it comes and it actually attacks the carbonyl group. Okay, and what they're not showing is you get that tetrahedral intermediate. And once we get the tetrahedral intermediate, just like always before, you get the collapse of the tetrahedral intermediate, and it kicks off the 3-thiol on the fatty acid synthase. Okay. This one does require NADPH. Okay, and we haven't seen NADPH very often. We talked about the one enzyme that could take NADH or NADPH, but for fatty acid synthesis, it requires a phosphorylated nicotinamide group. All right. Then you get dehydration and then reduction once again. Okay, and we're gonna don't worry, we're gonna see this and blow it up and just. And the structure that I'm going to show you in just a moment, it's very complex. The porcine, which is the pig version, which is very similar to the human version, it's been crystallized for most of the parts. So there's at least 1 or 2 portions of it that hasn't been crystallized to date because they're just too random. They're not structured enough, which means that they're probably moving, probably really active. And all of the active sites for the mammals are in different domains on a single polypeptide chain. These aren't multiple proteins that come together as a big complex. This is a single huge polypeptide chain for mammals. This is the part that's been crystallized. Okay, and I do want you to know the different domains. So there are 6 domains of a single enzyme here for us. This one is, once again, from the pig, but it is similar to ours. And for those who aren't color blind, you could see the different regions, and we'll talk about what each of these are. And as long as you know your enzyme names, they should make sense. So, for example, the beta-keto-acyl ACP synthase, what do you suppose it's going to be making? Exactly, it's going to be making beta-keto-acyl ACP. The transferases are going to be transferring the Acetyl-CoA to the malonyl, so on and so forth. And so that's why it's so important to know your enzyme family names. The only one that has a really generic name is acyl carrier protein. And then the special one, instead of thiolase, we have thioesterase. Whoops. Let me go back. And thioesterase is the part that actually will release it at the very, very end, whenever it's as long as it needs to be. Okay, and it's in acyl carrier protein and thioesterase are not pictured here because they were not able to be held down long enough in order for it to occur. And once you see what the acyl carrier protein does, it makes sense because it's the part that's the arm that swings everything around, or like the swiveler. So that is all one protein. Yeah, and there are 2 more domains that are not even. Yeah, it's huge, huge. It's a thing of beauty. Okay, this is the acyl carrier protein. As I mentioned before, in bacteria it's usually separate, but in us it's part of a domain of the very, very large complex. And so if we just break it down in parts, we've actually seen something like this before. So it has an amino acid side chain that looks like this, that gets activated by pantothenic acid, which is a cofactor that you need to get from your diet. And so what amino acid do you suppose right here could this be that has been activated that actually has the pantothenic acid attached to it for the acyl carrier protein? What is that amino acid? It's serine. Okay? This comes from vitamin B5, which you don't hear about very often. And at the very, very end of this, really, essentially, it's just a long arm. It's got that reactive thiol group. And so that thiol is both a good nucleophile and a good leaving group. And so that's the reason why it can take the acyl or an acetyl group or whatever and take it and then carry it, move it to the next part, move it on. We've seen this kind of biomolecule look like the end was different, but the reactive portion of it was very similar to something before. Oh, it's frozen up. What was that? What does it look an awful lot like? It's like that arm with the little reactive thiol at the very end. It does something very similar to the acyl carrier protein. In fact, sometimes I accidentally call it by the wrong thing.
Is it CoAsh?
Yeah, CoAsh, coenzyme A. It has fancy thinking. It has like the beta ethanolamine, and it's got the A portion, the adenosine, but otherwise it's very similar. And once again, coenzyme A is for the transferring of acyl groups. Okay, and we've already answered these questions. So don't worry, I've got this blown up. This is the spiral, which would be the antagonistic process as compared to beta oxidation. And so it's this little gummy worm looking thing. That's what it kind of reminded me of. It's a gummy worm with all the different parts. And remember, the parts that weren't crystallized are the thioesterase and then the acyl carrier protein. And as you can see here, the acyl carrier protein literally just picks it up, takes it, it's like the little robotic arm that just takes it from subunit to subunit to subunit. All right, so once again, before we can even start with step one, we've already made the malonyl intermediate, and we have to have an activated complex that can occur. So what we see here is the malonyl acetyl-CoA ACP transferase, which it tells you exactly what it's going to do, is it transfers the acetyl-CoA to the ACP. Okay, so it adds on this acetyl-CoA portion, frees up the coenzyme A, and just makes it an acetyl group, and then the acetyl group then will go ahead and transfer it to a reactive thiol on the K-S domain, which is going to be keto-acyl ACP synthase. Okay, so what do you suppose is, once again, the keto-acyl ACP synthase is going to do with an acetyl group? What does a synthase do? It's going to synthesize, and what will it synthesize ultimately? Beta keto-acyl ACP. Okay, so this is where I said possibly the -1 step, the malonyl group, actually gets to come in, and we get the free coenzyme A once again. Okay, so the transferase transfers the malonyl group to the ACP. Okay, so this is sort of like the loader. First, remember, it loaded on the acetyl group, then the ACP carried it to the KS. Then it's loading on the malonyl group. It holds it out over the acetyl group that's going to be added, and then we get that Claisen condensation type of reaction, where here, maybe if it would COCH2 carbonyl, so that way I can draw it out. Oh, I don't know if this will show up on yours or not, but we'll find out. All right, so what we have here, of course, that's just a carboxylate. So we get the electrons to smack down to the carbon. Let me switch to a different color. Then the electrons that were between the carboxyl group and the neighboring carbon, which would be the alpha carbon, if you remember from organic chemistry, because here's where the chain that we're adding it onto is. It's going to break and swing down and attack the carbonyl group of the acetyl, the acetyl group that's being added. Okay, and so here's the methyl from the acetyl group. Of course, this is coenzyme. Oh well, I was about to call it coenzyme A. This is that's on the previous subunit. Okay, so something has to break or give. What breaks is going to be the form of the tetrahedral intermediate, the carboxyl group, like what we see in organic chemistry. And then, I don't have enough room to draw it, you get collapse of that, and what leaves is the KS domain, the keto synthase domain. Now it's free. It's got the free thiol to wait for the next round. Okay? And so now the acyl carrier protein has a 4-carbon containing unit on it. That's why it's not called the acetyl carrier protein. It's an acyl carrier protein, which means it just needs an acyl group. So it's going to grow and get really big. The growing chain is on that arm. It's going to go from subunit to subunit. Okay. Yeah. Oh, awesome. All right, so that's a Claisen condensation reaction, if you remember that term from organic chemistry. And so now the acyl carrier protein has 4 carbons on it instead of 2.
What is the KS telling us what
It was the,
subunits were?
well, the KS, this one up here, is the enzyme subunit that allowed for the synthesis to occur for the keto acyl group. Because now, if you notice, the protein, no, not the protein, I'm sorry, the product that we have here is a beta keto, well, acyl group. Okay, so here is the beta keto group. Oops, this doesn't match up on mine. There's the beta keto, because there's alpha and there's ground 0. And so that's an acyl. Now, some people get confused because I don't know why the book does this. It gives it the very specific name, ketobutyryl, but that just means 4, because then it's going to be hexyryl when it's 6 and octyryl when it's 8. That's why the enzyme is just called ketoacyl, because it doesn't have a specified number.
The ACP is basically like swinging that group around.
Right, it's like your hand. It's like, okay, I'm holding on to it. Okay, and it swings it over to, if Mike was the KS domain, he adds on the acetyl groups. Now I've got 4 on it, and so now I'm going to swing it on, because we're trying to make a saturated fat, so we've got to get rid of the oxygen. So I'm going to swing it onto the next, subunit, and which would be, I guess, rylin. So we want to get rid of that double bond, because remember, we're essentially trying to store the energy, and you store it in the form of the higher degrees of saturation. The higher the oxidation, the more energy that's already been spent out of it. So we're trying to get rid of that oxygen. Where did I just throw? Oh, there it is. My stylus. So here's the second one. It's a reductase. Okay, and it's the KR domain, which stands for keto acyl ACP reductase. And so anytime that we're reducing, what happens to the number of carbon to oxygen bonds? Do they increase or decrease if you're reducing it? It decreases. So it goes from a carbonyl or an acyl group to an alcohol. Okay? And this requires NADPH. So once again, you want to pay attention to the cofactor here. So now we have a beta hydroxy acyl ACP group. And if it was 4, it's a hydroxybutyryl, but don't pay attention to the butyl part. That just means 4. Once again, that's only true for the first round. The second round is going to be 6, and the third round is going to be 8, and then 10. So the KR is for keto acyl ACP reductase. It tells you exactly what it's doing. It's reducing that ketone to an alcohol. But notice once again, it's still attached. So my hand, the ACP protein now, after rylin is done making it to an alcohol, shame on you, rylin, making alcohol, is going to swing it on to the next one. So very similar to beta oxidation, we are going to dehydrate it to make it into a trans fat. So remember that I mentioned that there are only 2 naturally occurring trans fats. One was in the breakdown, during the breakdown of the fatty acid. And remember, it doesn't ever leave so you don't have to worry about it clogging up your arteries if you're healthy, because it's the only naturally occurring one. And then the second one is during the building it up. And once again, as you can see here, the acyl carrier protein still is holding on to that trans fat, so you don't have to worry about it doing something erroneous. And so this will then make the enoyl ACP, technically the trans-enoyl ACP intermediate. And so that's why it's the dehydratase. Okay. So that step is very similar to beta oxidation. So what's the last thing that we need to do? We've got it as a double bond. So if it was to leave now, it would be an unsaturated fat, but we're trying to make it into a saturated fat. So what needs to be added? Hydrogens. And so that's just another way for a reductase. And so this is the enoyl ACP reductase. So once again, my hand, the ACP protein, still holding on to the trans fat, not going to let that little booger go. And then it goes and it moves it over to the enoyl ACP reductase, the ER, which once again utilizes NAD pH in order to make it the saturated fatty acyl group. Okay, so now we started off with just had 2 carbons, made it to the malonyl CoA, which had 3. Then we started we add it in groups of 2 at a time. So this is 4, the next round would be 6, then 8, then 10, then 12. And so it just goes on and on and on again. So it spirals until it gets to a certain length. Okay, so that's why that little acyl carrier protein just takes it around the different the different domains, the different regions of the FAS. And then, for whatever reason, they're still investigating this, that once it gets to palmitate, which is 16 carbons, it stops. Okay, and thioesterase, which is the TE domain, which was not shown, it just uses water. And so it's just the water is a nucleophile. It goes, it smacks the carbonyl group, and it frees up the fatty acid. Okay, so now we made our fatty acid. And so just to make that palmitate, it requires 7 ATPs to activate to make the malonyl group. And then it's going to require 8 acetyl-CoAs and 14 NADPHs. And so,
Would you expect us to draw like the cartoon version with that protein?
I mean, yeah, or just say like the TE domain is the thioesterase, or KR is ketoacyl, ACP,
Like
reductase. Right, just to know that. Yeah, don't try to draw the gummy worm. I can't draw the gummy worm. All right, so that's compartmentalization up to 16. Remember, so for us, and I'm not going to expect you, because we don't have the time to go into plants, and since this is for more of the biomedical thing, we don't go into the plant chapter of the chloroplasts. So I just expect you to know for animals and yeast that it occurs in the cytosol. Okay, and that's also where the NADPH is made. So the pentose phosphate pathway, and we're going to find out there's 2 ways to make NADPH. Primarily the pentose phosphate pathway, which we've already talked about, and then there's also an enzyme called the mammalian enzyme in a moment. The early stages of steroid, or let's just say sterol and isoprenoid synthesis can occur here, and this is where fatty acid synthesis. The specializations occur in a couple of other places. One is for phospholipids and steroids for anything longer than 16 and to make the desaturated, so we're talking about like the PUFAs and things like that, then that's going to take place, for us it takes place in the ER. And then the mitochondria does do some elongation and some, we already talked about ketone body synthesis. Okay. But for up to 16 carbons, it's going to occur in the cytosol. Okay, so fatty acid synthesis occurs in the cytosol for most of organisms, including us. Where have we been talking that acetyl-CoA normally is? Pardon? Oh, right, mitochondria. I thought you said something about the conch. And so, yeah, mitochondria. Okay, so that means acetyl-CoA cannot pass through that holy of holies. So it's stuck inside the mitochondrial matrix. But we have to have a pool of it outside in the cytosol as well in order for us to be able to make the fatty acids. So these are 2 separate pools of acetyl-CoA. And so the coenzyme A, we have to have it in 2 places. And so we have to talk about the effective way that we can transport acetyl-CoA from the mitochondria to the cytosol and back and forth. And it's a little bit more complicated than the glycerol phosphate shuttle that we talked about in the past. Because that was for the transfer of electrons, effectively to transfer the NADH from the cytosol to the mitochondrial matrix. Okay, and so this is just to show you once again, acetyl-CoA is impermeable to the inner membrane. So there must be coenzyme A there's a pool of coenzyme A out here. There's got to be a pool of coenzyme A within the cytosol as well. Okay, so to start off with, we can take acetyl-CoA, transform it to citrate using an oxaloacetate with citric synthase, which was the first step of what pathway? Citric acid cycle. And then the citrate's got a transporter, and so that way it can just go right on out. Okay. Okay, so now we have a new player, a new enzyme. I've already given you the answer. It's citrate lyase, and it's the one that requires an ATP and coenzyme A. So it's going to utilize a coenzyme A that's on the cytosol. And effectively, now we have acetylated it, so now we have an acetyl-CoA out here, which can go on for fatty acid synthesis if we need it under those conditions, okay, which presumably we are since we're shipping it out. So the byproduct for this is oxaloacetate. And oxaloacetate is going to use up an NADH to be transformed to malate. And so what enzyme does that? Malate dehydrogenase, technically be the cytosolic versus the mitochondrial version. And then there are two possibilities for malate. One is what we've seen in the past. Malate can just go right on through. Okay, join right back up to the citric acid cycle, or even gluconeogenesis in the event that we are under those conditions. But the second possibility is the second way that we have to make NADPH. And that's when the malate gets decarboxylated to form pyruvate. Okay, so we have a couple of ways of going from oxaloacetate to pyruvate. One is through gluconeogenesis with PEP carboxykinase and the pyruvate carboxylase. The other way is via malate and the malic enzyme. Okay, so if we need to make NADPH, you're going to use malic enzyme. If you need to store energy in the sense of glucose, then you're going to go through gluconeogenesis.
Is that the name, malic enzyme?
Yeah, it's really generic. Okay. And actually, we've already talked about this where. So, once again, there are two possible ways. Pentose phosphate pathway, and then now we have malic enzyme. All right. The pyruvate itself can then go back and forth, which we discussed, because there is a transporter. And so once the pyruvate is there, then we have pyruvate carboxylase, which is, can convert it back to the oxaloacetate. And we've seen oxaloacetate be utilized in multiple pathways now. That's why they're very intricately related. Okay. So let's talk about regulation. And I want you to be able to understand why it makes sense. Okay. So let's start with citrate lyase, which was the new enzyme that we haven't seen in the past, the one that's going to make acetyl-CoA. Insulin activates it. Why does it make sense that insulin will activate the acetyl-CoA for the storage of, or the synthesis of fats? When do we have insulin in our system? If you're healthy, once again. Glucose is high. So what's an instance when glucose is high? You've just eaten a meal, so now we're going to have to store all the energy for future use. So you have your energy-rich meal. Insulin will help trigger the fat stimulation. It's one of the reasons why, if you're trying to lose weight, you're supposed to try to keep your blood sugar relatively stable. Because if you go through the swings where you starve yourself, then you eat a meal, your body will kick in and think, okay, I've got to go ahead and store this fat for later. So it's self-defeating. Citrate, actually, if you have a lot of citrate around, it's also going to stimulate this process to make malonyl-CoA. Under what circumstances will we have extra citrate? This one's pretty easy, but when do we have extra citrate? Where's the citrate coming from otherwise, other than this process? Citric acid cycle. So why would it start to accumulate?
When the body doesn't go through the citric acid.
Right, we don't need to go through citric acid cycle because we've got plenty of energy. So, and now the ways to turn it, to downregulate it is, well, glucagon, which makes sense because glucagon is the antithesis of insulin, so I don't know if you want to consider the Lex Luthor Superman or Superman to Lex Luthor, whichever floats your boat. Epinephrine, why would epinephrine be a phosphorylation, which we still have 4 million people. Why would epinephrine do this? Under what circumstances do we need epinephrine? You're running from the bear, which once again, if you're running from a bear, you need to go downhill. Okay.
Or down speed bump.
Yeah. Okay. And then if you have an excess of Palmitate-CoA, it's going to downregulate the carboxylase as well. Why would we have an excess of Palmitate-CoA? What is it the product of? Fatty acid synthesis. So if we got we just made lots and lots and lots and lots of fats, then remember that's the 16. So that's probably the only one that I'm going to have you memorize the name of for the common name, because palmitoyl is 16. But it's going to turn off or start to downregulate it if you're healthy. So, yep. Are there any questions? We ended like right on time. I'm impressed. Dr. Kimball should be impressed too. So, if there are no questions, then I'll go ahead and I'll end class with prayer. So, generally, Father, I want to praise you and thank you for this wonderful day and for the beautiful sunshine outside. And we're so honored and blessed to be able to gather together and work and study in your name. I ask that you reach out and take, for those of us who need a touch in our body, and you see Hannah and Mariah both, and we know that by your stripes that we are healed. In Jesus' name I pray. Amen. What?
I was wondering why I didn't hear anyone else say anything.
I know, yeah, no, she's, all right. Dr. McConkey sent her home in histology, I guess, or, yeah, histology?